Okay. Hi guys, I'm Holly.
I'm Deanne.
And I'm Ria.
And our Ignite talk today is on human microsatellite DNA, as you can see. And this module will mostly remind you probably of a CSI scene. And we are the principal investigators of the Budgerton case. So Sadat is out there alive and well today, so he was not harmed in the making of this presentation. Okay. One of our dear Bio 1500 classmates, Sadat, was found dead in RPCC this morning. We saw red splatter stains on the carpet and a shattered Snapple glass near the scene of the crime. We investigated the incident, interviewed staff and students, and then we narrowed down the murder to only 3 suspects. Those people are Mario Carrillo, our very own Dr. Mark Sarvary, and Kelsey Scalar.
Okay, so with the 2 biggest evidence, we decided to carry out the presumptive test for forensic analysis. And we first did the Kastle-Meyer test to test for blood. So for this, we put ethanol on the swab, then swabbed it against the red blotch, as you can see in the picture. And then we put ethanol, the Kastle-Meyer reagent, and then the hydrogen peroxide. And when we did this test, our swab turned pink, which means that blood was present. And for the broken glass rim, we did the amylase diffusion test to test if saliva is present. And this is when we put the agar starch gel and iodine solution and put the sample in it. And if there is like a blue stain, it means that starch is present and amylase is not. And if there's a clear circle, it means that amylase is present because it broke down the starch. And when we did when we did this test, there were no clear circles, so it means that saliva was not present. And with the DNA found in the blood, we decided to carry out PCR to amplify the DNA. And to do this, we needed DNA polymerase to synthesize the reaction, the original DNA sample, the DNA primers, which are complementary to the flanking sequence, the 4 deoxyribonucleotides, and heat. And the heat was applied at different temperatures depending on the conditions. So first, it was at 90 degrees to denature the DNA, and then it was decreased to about 50 to 60 degrees to allow the primers to attach, and then it was increased to the optimal temperature for the DNA polymerase. And this is often done with multiple primers and at multiple loci to save costs and time, and this is called multiplexing.
So either a gel electrophoresis or a chromatogram can be used to evaluate the results of a PCR. In our case, we'll be using chromatogram. This example shows a chromatogram of the 3 out of 10 loci from our PCR. A locus is a specific position on the chromosome. The peaks shown for a single DNA donor will either be homozygous with a very large peak or heterozygous with 2 smaller peaks. Each peak will have a fragment size that will be shown in the bottom. In this case, we have 281 and 285. Now, what do we do with these numbers? Each fragment size includes a flanking sequence, primer length, and base pair repeats. And this is the general formula. So for our example of 281, we are given the following information. From there, we just plug and chug the values, and we get an allele number. To use this allele number to find the genetic profile frequency, we use the allele frequency of the population. Then we plug in the value to a genotype frequency formula, which are different for homozygous and heterozygous alleles. We then multiply these genotype frequencies to get a random match probability. Finally, we take the inverse of that, and we get a genetic profile frequency. This tells us how frequently the exact same genotypes for these alleles will appear in the population. So the more loci we look at, the more we can narrow down suspects because there is a smaller chance of 2 people having the exact same genetic profile.
Okay, so in a real-life forensics case, only analyzing the 3 loci shown above is not a really accurate measure of determining whether or not you found the real murderer, but for our case here, we're just going to look at these 3. Just think about it's more likely to have 3 loci common with like someone sitting next to you than it is to have 10 or 15 or 20. So just by looking at the chromatograms here, it's up to you guys to determine who the murderer is. I'll guide you along a little bit. So the first sample up there shows the tested sample, which means it's from the swab that we took in RPCC this morning. And then the next 3 chromatograms are samples from our suspects in the audience who may be the murderer. The easiest way to see what's going on is to find the chromatogram that is the same or is most similar to the tested sample. Okay, so now we're going to have a poll everywhere question where you can actually input who you think killed our dear Sadat. So these are the three choices. We'll give you a few minutes to vote. Okay, so based on the conclusion drawn by all of you, it looks like...
I just changed it so students can see who's voting what, but give them another 30 seconds.
Okay.
Did everybody vote? Okay. You want it back in the pressing?
Yeah. Well, so it looks like Kelsey Sklar may be the murderer of Sadat, but as you know, we've only presented analyzing the 3 loci, so that's not like going to actually tell you this is exactly the murder from the DNA profiling. And so here's the moment you've been waiting for, who actually did kill Sadat. You're right, it was Kelsey and not Dr. Sarvary for Sadat making bad jokes during his lab practical last time. So mostly we'd like to thank all of you for participating in our lecture, and we hope you learned something new about DNA analyzing and microsatellite DNA. Also big thanks to Sadat, Mario, and Kelsey for volunteering for this.